So before we start, I would like to talk about the anatomy of the plant cell. And for the IMAT and BMAT exam, I didn't actually find any questions that asked on the anatomy of the plant cell as a whole plant. I did, however, find many questions when it comes to the anatomy of the cell itself, the cell anatomy. So I brought here a diagram for the general structure of the plant, but I never saw any question that actually asked about the following structure. I mean, you can understand and if you want to memorize the following structure, but just know it's very low yield to know the actual plant anatomy. What is extremely high yield is to study the following structure. The following structure is called the chloroplast. The chloroplast is an organelle that you can find inside the plant cell. So we talked about the fact that you have plant cell and they have a cell wall which helps them to be square and not animal cell cycle, a circle. And inside the plant cell you have many chloroplasts. And this chloroplast is basically the following structure. So I'm basically zoom in on one of the following chloroplasts. So I'm going to mention in this video the highest yield structures you need to understand inside a chloroplast and the overall idea of what is a chloroplast. So a chloroplast is the place where we do the light and dark reactions. The light and dark reactions are basically the autotroph part of the plant. It allows the plant to create food for itself in a source of carbohydrates. So the plasma inside the chloroplast, the area between all the parts, is called the stroma. Inside the chloroplast we have DNA. And if you remember, what other organelle actually contains DNA? The mitochondria. And the chloroplast and the mitochondria are very similar also in their chemical reaction we are going to see in a minute. So we have the chloroplast DNA. Then we have a stack of something that looks like a lot of chip, and this stack of a lot of weird things is called the granum. The weird things that basically compose the granum are basically stacks of thylakoids. Many thylakoids are going to give you the granum. So it's thylakoid give you granum inside the stroma. Now the chloroplast contains multiple amount of membranes, which is important to remember. And it also contains ribosomes, of course, because we need to create proteins from the DNA itself. The proteins are usually cell membrane protein that are going to help us to create energy, which you are going to see in a second. Very similar to mitochondria as well. I would like to zoom into one of the thylakoids. Inside the thylakoid we are also going to have another membrane. And on this membrane we are going to have very important proteins. This membrane is called the thylakoid membrane. Inside the thylakoid you have the lumen. And inside the lumen you have a very important process. Do you remember in the mitochondria we used and we made the protein gradient, the proton gradient, in order to create the electrochemical gradient? We are going to use something very similar here. In the next video you are going to see that on the thylakoid membrane you have different proteins. And these proteins basically can absorb photons in the sun. They can yield the sunlight and they can cause the excitation of electrons inside this protein and the structure, the chlorophyll, which we are going to study in the next lessons. And basically once the electrons go back to their stable state, they release energy. So photons are basically going to excite electrons, and once they go to the back state after they are being excited, because they always go to the most stable state, they release energy. But this time we are going to yield the energy very similar to the electrochemical gradient and the complex of the electron transport chain. We are going to yield the energy to pump protons to the inside of the lumen against the concentration gradient. So we yield sunlight and we are going to have ATP synthase, very similar to the mitochondria, which is this time going to pump protons to the inside of the thylakoid. And now once the protons going to follow out, they are going to follow through the ATP synthase, and this time it is going to yield energy to create ATP, and we are also going to yield energy from this proton gradient to charge NADP+. How this process actually occurs, the charging of NADP+ and the ADP to ATP through the ATP synthase, we are going to study in the next lesson. In this lesson, I just wanted to show you the most important parts that we actually have inside the plant anatomy, inside the plant. Because while it's very low yield to study for the IMAT and BMAT exams, the following plant anatomy, it's very high yield to study the actual chloroplast structure for the exam. There are several more parts of plant anatomy that you should learn, and I will mention them in the next videos, but know that it's mainly involved the microanatomy and not the macroanatomy of the structure of the plant. So if I will sum this video up, inside the animal cell, you have something called the chloroplast, the plant cell, You have something called the chloroplast. You also have mitochondria, but you also have chloroplast. The chloroplast allows us to create the carbohydrates from sunlight. It also allows us to get ATP and NADPH, which are basically going to charge the dark reactions, which the dark reactions is where the actual carbohydrates are going to form. So it is divided into two steps. The first step would be to excite electrons in a specific structure inside these proteins that are called PS2 and 1, which we are going to study in the next video. And the excitation of these following molecules, the charging of them using the power of the sun, is going to excite electrons that are going to found inside the structure. And once they are excited, once they are going to go back to their relaxed state, they are going to release energy. This energy is going to be yielded in order to drive protons against the concentration gradient. Once you have a lot of protons inside the thylakoid lumen, you are going to have the ATP synthase, which is this time going to let and allow protons to go outside of the lumen. We are going to yield the energy to create ATP, and in different processes, we are also going to charge NADPH to NADP plus to NADPH. I will show you exactly how everything occurs in the next video.